Great. Thank you very much, Joachim, for taking the time to speak to me today. It's great to catch up with you. Please, could you describe your role in Bonn?
Yeah, thanks for having me. So I'm a professor for genomics and immunology, basically in Bonn. I have been at the Bonn University for a long time. And since 1st of September, actually, I started to develop a new section at the German Center for Neurodegenerative Diseases, which is called Systems Medicine, and that's my future focus.
Thank you very much. Can you please tell us a little bit more about your work and current priorities?
Yeah, so we have basically invested into single-cell omics technologies for quite some time. And for this purpose, we also developed a platform here in Bonn, which is called the PRECISE platform for single-cell genomics and epigenomics, which is actually a joint venture between the university and the German Center for Neurodegenerative Diseases. We joined forces because the technologies are evolving very quickly, and we thought it would be better to do that together. The idea we have here is to use single-cell omics technologies in different flavors, single-cell RNA sequencing, but also single-cell chromatin analyses, and so on, to better understand diseases. So we're really focusing very much into bringing these technologies directly to patients in human studies, explorative, but also in the meantime, we have started to run single-cell omics in clinical trials.
Thank you very much. You are heavily involved in single-cell omics and also bringing this to the clinic as well as data analysis. So what exactly this means to your group?
Yeah, so the challenge, of course, is you have a very new technology, and if there are commercial products, we're still learning how to use this technology. In the lab, it's probably easy to do an experiment where you have very few samples from an animal model or a cell line also. But if you then go into the clinical setting, new questions arise. You have to have standardization. You have to organize that you get the material in time. If you use biopsies, then you have to work hard on identifying what are the best conditions to get cells out of the biopsies. Sometimes with certain technologies that are often used, you lose spatial context. So you also have to develop new ways of measuring the spatial context, which, for example, is in spatial transcriptomics. So you have to also develop and establish new technologies. That's one large challenge that we are basically facing and that we are addressing. And then, of course, when you have more samples from clinical studies, we have complicated data in single cells. And if you have more individuals, then new mathematical and statistical questions arise that have to be addressed in a very complex data space. And so we're also here encountering new challenges that we then have to address. Very often we do that in collaboration with experts in statistics, in machine learning and data sciences. But what we also have done in the meantime, and this is what is the impact on our group, is to include into the group people from very different disciplines. So we have really medical people, physicians, but we also have data scientists, molecular biologists, people from molecular biomedicine. So it's a very diverse group of people with very different expertises. And only if you have this kind of team, then you are able to bring these technologies really to the clinic. So this is an area where, the old days of science where you were in your lab and you did an experiment and you found out something, they're really over. So we really have to have teams with many, many different expertises, and they have to jointly work on them. And so skills are not only scientifically, but also how you communicate, how you interact with people, and how you learn to understand people with different background, which from their science have different languages even. So this is the kind of things which we have to establish. And I think we're on a good way in our group.
That sounds very interesting, Joachim. Thank you very much for sharing this. I can only wish you good luck for this. What are some of the major challenges that you are working on overcoming right now?
Yeah, so single cell analysis really is a discovery technology, and you can find out new things. And of course, we are in the middle of a pandemic, and we have a new disease. And so what we actually did, in addition to our programs on Alzheimer's disease, on chronic obstructive pulmonary disease, and in other infectious diseases, we decided early on in February, when basically the first signs were there in Europe, we decided that we will contribute to a better understanding of the COVID-19 disease. So we built a program and started bottom up. We built a network, actually a national network, which we call the German COVID-19 Omics initiative. Yeah, so which basically brings together experts in genomics technologies, NGS technologies, and data analysis. And we teamed up with many others who have access to patients, colleagues from the intensive care units, from the outpatient clinics, so that, again, this teamwork to basically being able to study the response of the patient's immune system to this virus. And since the immune system is very complex, we decided to use single-cell omics to do this. So we applied our knowledge, our expertise, and our technique directly to patient samples. And what we could identify and find out by this is that we basically have found that in these severe patients, so with severe COVID-19, these patients have a completely different immune system response compared to those that have mild versions of the disease. And that's important because with these findings, we also identified cells that we can actually try to reprogram. These kind of immune cells with similar functionality have been seen in cancer when you have a situation where the immune system doesn't fight the cancer anymore. So these are suppressed cells. And we find these also in these COVID-19 patients, in the severe patients. And if we know all this, and we can use this knowledge basically to design new trials to test drugs that might reprogram these cells. And I think that's the important thing, to find out what new diseases are, and then quickly use this information, this molecular and cellular information, to trigger new therapy trials and find out which drugs could work. What's interesting, early in April, we already saw from these kind of analyses that the dexamethasone is probably a good drug candidate. And that was a time when this was, discussed by some clinicians, but others were finding this idea not so good, to say. But we could see molecular patterns in these data that we generated that clearly indicated that certain patients with severe disease should actually benefit from treatment with dexamethasone. And in the meantime, trials have been performed with this drug, and it's very clear that severe COVID-19 patients can benefit from this drug. So I think it's very important to bring these new technologies directly to patients, not only in this crisis situation with this disease, but also with other diseases where we still don't know exactly what the molecular and cellular mechanisms are.
Great, thank you very much for sharing this with us. The current coronavirus pandemic has altered the priorities of many researchers. Have you shifted your research towards projects or areas that are of direct relevance to COVID-19?
Yeah, so as I said right now, we really use the resources that we have and the knowledge to really gain information about the disease. And so we have started this large program, and we continue to do that. Yeah, so it's important because we also can demonstrate with this disease how much information can be provided by these new modern single-cell technologies directly to understand disease. So it's for us not only that we contribute to help to overcome this crisis faster and find ways to treat patients with severe COVID-19 better, but it's also a means of establishing systems and organizations and administrative processes that allow us to faster apply single-cell technologies to clinical trials, to apply them to patient samples in a much more forward way. And so, honestly, although, we're all in this crisis and we all want to have this, of course, that it goes away quickly, we also took the opportunity to say, like, can we learn under these conditions much faster how these new technologies can be brought to patients. And so I have a positive feeling and a negative feeling about the pandemics. I, of course, as everybody suffers from that, but on the other hand, we learned a lot. And it expedited many things that we had planned anyway, to do that much more quicker now. And that's at least in this situation a good thing.
Thank you very much. This sounds very interesting. The single cell analysis market has seen exponential growth over the past years. In your opinion, what are the key factors driving its growth?
Well, the key factors are actually that these technologies are revolutionary, because now we can see cellular mechanisms and cellular behavior in an unprecedented way we have never been able to do before. And this is for the first time, we have the capabilities to look on the unit of life, the cell. We can look at the cell in a such deep manner that we can now identify and discover things that were just not possible with techniques before. And so I think, we will first of all learn much more and much deeper about the processes of life itself because of these technologies. But I think even more important, we will learn to describe and then to understand the real processes between cells in diseases. Yeah, and that's something, many of us in many disciplines have started. If you think about immunology, there are technologies where you look at single cells, for example, flow cytometry, but you usually measure in a very biased way. You can only measure certain parameters that you have to define beforehand. And of course, that brings in bias, and that also brings in, of course, very often discussions about the interpretation of these data if you're not using the same parameters in different labs, and then you have difficulties to compare them. There's many examples of that. So these analyses are on single cell. You measure the molecules per cell, but since you're not able to measure the complete space of data in these cells, you're biased. And these new single cell technologies based on omics technologies take that completely away, because now you can measure in an unbiased way, and the better the technologies become, the more parameters you can actually measure and then analyze, and that changes the whole thing. And that's why I think that's the key driving factor. And in the meantime, many people and also, of course, companies and industry has understood that this is a revolution. And the more players are coming into the field, applying these things to important questions in medicine, the more this will actually grow. And that's why I think we're seeing an exponential growth at the moment. It's very exciting. It's and many of the early challenges we have been able to deal with, and that's why also there is much more confidence now, because there have been points that people were thinking cannot be technically achieved, but they are now achieved. So hurdles in front of us don't look as high as before anymore. So I'm also one of those that are very optimistic and confident that we can really, I would say in the next 5 to 10 years, bring these technologies really to a clinical practice even.